terrestrial habitats, as well as I'll try to talk to you about the different kinds of methods that can be used in assessing biodiversity. Okay, so looking at your syllabus, I'm sure that you already know what biodiversity is, what are the types of biodiversity, what are the different kinds of biodiversity. But I'll just go through it in a few slides, and I'll just continue into the different ways in which we can assess biodiversity, and what are the different ways in which we can understand what biodiversity is present in aquatic habitats as well as in terrestrial habitats. We'll also try to understand why biodiversity assessment is important to us, why doing it is important, and then we'll try to end with one example from my own study. Okay, so I'll just move to my slides. Okay, right. Now, as I told you, everybody knows what is biodiversity. Biodiversity, by definition, it means that it is variety of all life forms. Variety means it's the different kinds of all the living organisms that are present on the face of the earth. Like we have microorganisms, we have plants, and we have animals. I'm a zoologist and I work on animals. So in animals also we have a lot of diversity, starting from animals which are unicellular to animals which are multicellular. Animals which live on the land, animals which live in water, animals which live in marine water, animals which live in fresh water. So there is a huge diversity of animals, plants, and microorganisms that you find on earth. So when we use the word biodiversity, we look at that variety, we look at that diversity. And all these organisms, all these living organisms, all these life forms, they work together. They are dependent on each other within the ecosystems like a intricate web. That means they are extremely well connected to each other, and this connection helps to maintain the balance, right? And also it supports life on earth. If anything goes wrong in any small component of this biodiversity, it may result in a disaster, right? And as you all know, biodiversity is important to us because everything that we get from the nature, everything that we get from our mother earth, everything that we need to survive, like food, water, shelter, and even medicine, it comes via biodiversity, right? Now, moving forward, there are three different kinds of biodiversity. The first one is species diversity, where we look at the different kinds of species that are biodiversity. It may result in a disaster, right? And as you all know, biodiversity is important to us because everything that we get from the nature, everything that we get from our mother earth, everything that we need to survive, like food, water, shelter, and even medicine, it comes via biodiversity, right? Now, moving forward, there are three different kinds of biodiversity. The first one is species diversity, where we look at the different kinds of species that are present in a given ecosystem, like species 1, species 2, species 3. We call this as the species diversity, right? And it is the number of or different number of species present in a given ecosystem. Like for example, if I'm talking about aquatic ecosystem, so the number of zooplankton, number of fishes present in that aquatic ecosystem constitutes its species diversity. Now, if you look at the second type, that is ecosystem diversity, here we speak about the different kinds of ecosystem within which this species diversity exists, right? So they include the various habitats, the various communities, and the ecological processes happening within these habitats. So this is a desert ecosystem. All the plants, all the animals, all the microorganisms, all the geological, biochemical processes happening within this ecosystem and freshwater ecosystem and all other ecosystem constitutes the ecosystem diversity. And finally, we have the genetic diversity, where we look at the variety of genes within a species. So here we look at species, here we look at ecosystem, here we look at the genetic information within a species or within a population. Okay, and this information has to be inheritable, right? And it should be within interbreeding population. Right. Now, so we can conclude that biodiversity includes species, biodiversity includes ecosystem, biodiversity also includes the genetic information. Right. Now, these three words, terms you must have heard many times: alpha diversity, beta diversity, and gamma diversity. Now, how do you define this? Alpha diversity means when we look at a particular habitat or particular ecosystem, say forest ecosystem here, and then we look at the species that are present within this particular habitat, we call this as alpha biodiversity. So, if I look at butterflies of a given area, I can call that as alpha diversity or alpha biodiversity. And if I look at the butterfly species of this particular habitat and I compare the butterfly diversity of this habitat and maybe this habitat, or just we'll just say these two habitats, we call this diversity as beta diversity. Now in alpha diversity, we look at just the species in a single habitat. Here we compare two habitats. So when we compare two habitat, we call it as beta diversity. And the overall diversity within all the habitats is called as gamma biodiversity, or we simply call it as gamma diversity, right? So alpha, beta, and gamma diversity. Most of us will be looking at this particular component. Few of us will try to compare these two sites. So maybe we'll try to compare two dams, or maybe we'll try to compare two forest, or maybe we'll try to compare two areas, and we'll look for butterflies, or we'll look for zooplankton, or we'll look for birds of that particular area. Now, biodiversity is important, as I told you, for healthy functioning of ecosystem. It is important to us economically. Fish, you all know, they give us food, they give us monetary benefits. Ecologically, biodiversity is important. Scientifically, what we are doing right now, what you as PhD students, I as a PhD guide am doing, is because of biodiversity. It also has cultural relations to it, and also recreational services biodiversity will provide to us. So if you must have visited a few parks, or if you must have visited Ramoji Film City in Hyderabad, there you will see a butterfly garden, and that provides us with recreational services. Okay, so as I told you, it gives economic or monetary value to us because of what we receive from the forest in the form of medicine, food, wood for construction, and for other industries, water, right? Everything we get from biodiversity, right? And it also helps us to maintain our cultural identity. So if you must have heard about the Egyptian culture, it was all because of the river Nile. And if you look at the river Ganga or Yamuna, we have our own cultural identities because of the biodiversity that is present in and within that particular habitats. And as I told you, it can give you recreational value, providing experiences to us, like we go for hiking, we go for mountain climbing, bird watching experiences, and we can also earn economic profit through a very upcoming type of business called as ecotourism. Right? Now the question is, why do we need biodiversity assessment? So what is biodiversity? Biodiversity is looking at what kind of plants, what kind of animals, what kind of microorganisms are present in a given ecosystem. Then we looked at three types of diversity: alpha, beta, and gamma diversity. Then we looked at why biodiversity is important to us. Now we'll have a look at why there is a need for biodiversity assessment. Now most of you will know that we are facing a great extensive pressure on our ecosystem. We are facing a lot of anthropogenic pressure on the species within the ecosystem, and that has resulted in declining of the biodiversity at a very rapid rate. And this is happening because of our direct influence on the biodiversity or the ecosystem, or it is happening indirectly because of our activities. Now if you look at the Anthropocene period, where we took over the earth, the extinction rate of higher taxonomic group and other organisms that include plants and animals as well has increased about 100 times. Right? Now this has happened because of our interference with the nature. Right? So one thing to remember is extinction of habitats, extinction of species is happening at an alarming rate, and that is happening because of our activities, and these activities can be direct or these activities can be indirect. And especially the higher taxonomic group like mammals, leopards, tigers, or amphibians, reptiles, they are the most affected, especially amphibians. If you look at amphibians, they are the most sensitive aquatic group, and whatever we do to their habitat, it results in their death or it results in crippling of those particular organisms. And this in turn has resulted in inadequate ecosystem services. Now, what are ecosystem services? It gives us food, right? It gives us medicine, construction, and other raw materials for us to survive. It helps in maintaining greenhouse gases. It helps in water purification, soil formation, pollination, very important thing that's happening right now, pollution remediation, nitrogen fixation. All these are ecosystem services. Now, what is happening because of the uh, decline in biodiversity? It is having a negative impact on our existence itself, right? And it is now resulting in significant economic loss. Now, for an example, if you look at beehives, so we spoke about pollination. So beehives, they are responsible for pollination. Lot of beehives are getting destroyed because of the extensive use of pesticides. And what is happening is, because we are losing these natural pollinators, we have to go for more expensive commercial pollination practices, right? That results in more economic loss. So what we are getting naturally, what we are getting for free, we are destroying it directly or indirectly, and we are spending a lot of money and incurring economic losses to get benefits out of mother nature, right? Now, declining biodiversity is the major reason that we should go for biodiversity assessment, right? Now, what do we do in biodiversity assessment? We look at extinction risk. We look at what is the status of diversity of a particular group of organisms in a given area or a given ecosystem, and we look at the trends in the population. Is the population increasing? Is the population decreasing? Is there any threat to the population? Is there any threat to the diversity? And we try to evaluate it, and then we submit a report to the government or to the NGOs to draft some good, efficient conservation policies and implement them to promote sustainable development, right? So the thing is, baseline studies, we call these studies we call as baseline studies. Baseline studies are important for proper conservation action plans. So the major thing to remember is, why do we need to do biodiversity assessment? We need to do biodiversity assessment to protect the nature. And why do we need to protect the nature? Because we are facing economic losses, right? Because we are spending a lot of money in getting what we used to get things freely, right? So it is for our own benefit. We need to go for biodiversity assessment so that we can protect the habitats, we can protect the ecosystem, and we can protect the species that are present within the ecosystem, right? Now, what do we do in biodiversity assessment? Now, as I told you, we also call this as baseline study. So here we look at the richness, here we look at the diversity, right? Here we look at the population, that means abundance of a particular species, and we look at heterogeneity of the species within different ecosystems. So the point to remember here is, what are we going to do in biodiversity assessment? We are going to look at the diversity, we are going to look at the population increasing or decreasing, and we are going to compare these species, or we're going to compare the populations with another type of same type of habitat. Okay. Now, we do it again for animals, we do it for plants, we also do it for microorganisms, right? Now, before we proceed further, I'll just go through some basic or important terms. The first one is richness, right? You must have heard about this word, richness. Richness means it is the unique species, or what we call as diversity, right? So if you look at this particular image, here you can see 6 different species within this given habitat, okay, or within this given ecosystem, right? So this becomes the species richness. Now, if you take out one species of this and you count the number of individuals of that particular species, we call this as abundance, or we call this as population, measurement of population, right? So what do we do in species abundance? We try to understand what is the number of species, individuals of species 1, species 2, and likewise we move to species number 6, right? When we do that, we are calculating the abundance. We are trying to understand the equitability, or we are trying to understand the evenness of the biodiversity, right? So species abundance and species richness. And what do you mean by heterogeneity? It means dissimilarity among the species in an ecosystem, right? Now we move to the actual point for today, which is called as biodiversity assessment, right? So biodiversity assessment can be done by 4 different ways. 2 ways most of you might be doing, right? The fourth way, few of you might be doing DNA barcoding or environmental DNA, what we call it, and many of you might be even going for, especially the people who are more concerned with plants, they might be going for remote sensing to understand the forest cover, land use patterns, or for mapping of habitats, right? So the first thing here we have to understand is look at is field observations, right? So most of the PhD students, most of the scientists, they will go out into the field and they will observe and record the presence of different species in a ecosystem. So for example, I work on zooplankton. I'll go to a water body, right? I will use my net. I will collect water. I will take that sample to my laboratory and I will observe under microscope, and I will try to understand what kind of zooplankton are present in my sample. Okay. So to do that, you need to use some methodology that is called a sampling methodology. So scientists, mostly terrestrial, in terrestrial habitats, we use different kinds of methods which we are going to look at today. Methods called as quadrat sampling and then transect sampling. Then if you're looking at tigers and leopards, we use something called as camera traps to understand the diversity as well as abundance of these particular organisms, right? Next method I told you, remote sensing, where we don't have to go there to that particular habitat. We can sit in our lab and we can understand what is happening to the biodiversity or to the ecosystem using satellites or using aerial imagery. Many are also using drones to understand what is happening or to cover forest fires, right? Then we have the most recent field, which is becoming much more advanced right now, such called as DNA sequencing to assess the genetic diversity. So if you remember, we spoke about species diversity, we spoke about ecosystem diversity, and then we have the genetic diversity within species or within populations as well. Okay, now we'll look at the first method. The first method is called as field survey, right? Where you go to a particular water body and you use different sampling methods to understand what is present in that habitat or in that ecosystem. So here you can directly observe the plants, you can directly observe the animals, you can directly observe the microorganisms or zooplankton that are present in this particular habitats. Now, to do this, you need to use some sampling methods or sampling techniques called as transect surveys, quadrat sampling, or in case of mammals, capture, mark, recapture studies. Okay, so one by one, what we'll do is we'll try to understand what are the different methods that you can use for terrestrial habitats, that you can use for aquatic habitats. Okay, now we'll first look at terrestrial habitats, right? Now, if you look at the terrestrial habitat, which is supposed to be one of the biggest ecosystem or habitats, there are hundreds of techniques or hundreds of ways in which you can look for richness, in which you can look for species abundance, right? We can, as I told you, we can divide them into observational techniques. We can separate them into capture techniques and also marking techniques. And this is the way which you can collect data. Okay, so these are all the methods which will help you to gather data, which will help you to create a baseline data, which will help you to give richness and abundance of a particular group of organisms in which you are interested, right? So first we will have a look at the quadrat method. Now before that, as I told you, observational methods can be direct, right? You just go to a water body, you have your net, you put the net into the water, you collect the specimens, you observe them, and you take them to your lab for further work. Or it can be indirect. Like for example, direct can be you can have a binocular and you can look at a bird through the binocular. That can be direct type of observation. Or maybe you can look at the excrement or scat of a leopard. Okay, when you're roaming through a forest, you have not actually seen the leopard. You have just seen its excreta, and by looking at the excreta, you come to a conclusion that there is a leopard in this particular forest, right? So this becomes the indirect way of understanding biodiversity, right? Now, what we'll do is we'll try to understand the first method, which is called as quadrat method. Okay, so quadrat method means using a square. Okay, so quadrat sampling is one of the most frequently used methods to study biodiversity and especially ecology, right? Now here what we do, we use something called as squares, which are also called as quadrats of equal size, right? So you have squares of say 1 by 1 meter, or you can even have in centimeters also. So 1 by 1 meters, we can have quadrats or squares, and you place them in a habitat of your interest, say a forest, right? So if you want to look at uh, organisms that are present in the grass, you will put the quadrat in the grass. So for example, if you're looking at plants, these flowering plants in the given area, so this is the given area, right? So there is one species of plant in this given area, right? So you take this, right, and you throw it into that particular area, place it into the particular area. The thing is, you have to place it randomly, right? And whatever organism or whatever plant comes within that quadrant fully, okay? So if it is half, you don't count. Fully, you count them and you measure the richness of a given habitat. Now, for what it is used? It is used mostly for slow moving organisms, right? Or non-moving organisms like plants, right? So quadrat, very commonly used. You use a square and you put it into a area of your choice. You place it randomly, and whatever organisms or whatever plants come within the quadrat, you measure it. Now you measure it monthly, you measure it weekly, you measure it daily. That depends on your research question. Okay. Now sometimes animals move, right? Sometimes organisms move from one place to the other. So if I place a quadrat here, right, and if I am looking at a butterfly, or if I am looking at a rodent, or if I'm looking at a mammal, I will not be able to catch it, right? So what you have to do here is you have to move from one place to the other. So what you do is you move in a line. So that is why this is called as the line transect method. So what you do, especially if you look going for birds, you start from here. Okay, so if you look here down, so you start from here, right, and you start moving from 0 meters to 100 meters. But the thing is, you do it on the same route every time you go for sampling. So if I'm doing it in the first week of every month, or if I'm doing it fortnightly, I will travel through the same place in the same line every time, right? So that you get repetitions, right? So as I travel, I'll start finding birds. I will note down the name of the species, and I'll also note down the number of individuals of the species that I found, and I'll stop here. Okay, so depending on your area, you can go for 100 meters or you can go for 1000 meters as well. So this is used for moving animals like mammals and birds. Okay, sometimes it becomes difficult for us to travel, right? Or sometimes it becomes difficult for you to move through a dense forest, right? So during that time, what you do is you don't move. You just stand in a particular place, or you set up your sampling station in a particular place. You have your recorder or you have your binocular, or you have your any other observing material, you sit there for a standardized time period and you record the calls of the bird, or you record the singing of the bird, and based on the call, based on the singing, or based on the observation, you note down what kind of animals or birds were present in that particular area. Okay, so this is very commonly used for bird, where you record these organisms from a single point, but for a standardized period of time, say for 2 hours, or say for 3 hours, say in the morning, or say in the evening. So this is called as avian point count. So the first method is quadrat method, where you draw a square. The second method is you draw a line and you walk through it. The third method is you stand there and you observe, or you record the calls or the singing of the bird, and then you can understand the diversity and the abundance of the bird species in a given area. Okay. Next is something called as spot mapping, right? So what do you do in spot mapping? This is a relatively intensive type of study. Okay, but the thing is, you cannot do it for more than few species, say 4 to 5 species only. You can look for it. So what you do actually here is you work on minimum number of species. You don't go for looking at the total checklist of birds or total number of birds in the particular area. You select the number of birds, less number actually, and then you search for them. You observe for them. So what do? You observe their activity, you observe their movement, you observe their interaction, and you do it frequently, right? So if you want to do a checklist of birds, you can go monthly, you can go fortnightly, once every 15 days. But if you want to do spot mapping, you have to go very frequently, say once a week, or maybe once every 3 to 4 days. And there you observe each and every activity of the birds in that particular area. So you know the territory of the bird, you know the territory of the species, and you be there for long periods of time, and you monitor each and every activity. This data can be used to understand the density of a bird in its territory. But the point to remember is this is extremely intensive. You have to spend a lot of time in the field. Likewise, you also have something called as nest searching, where you look at the nest of the bird. You actually go and search for the nest, and you look at the reproductive success rates. Okay, so you search the net and you nest, and you monitor the reproductive success rate by looking at the eggs, by looking at the way the eggs have hatched, by looking at how many eggs have hatched, how many have not hatched, if the clutch has failed, why it has failed. Again, like spot mapping, this is also a very labor intensive way of studying biodiversity. That's why, again, you have to look for very few number of species, but during restricted time, say during only the breeding season. Okay, and you monitor it very frequently. So this is called as nest searching. Now, next method, which we normally use for medium to large sized organisms, is called as the foot track survey. So if you go into the forest where there is no tar road, normal soil is present, you can go there and you can have a look at the tracks left by the animal. So as they travel from one place to the other in search of food or mate, or they are hiding from predators, they will cross a particular predefined path. So this is the path in which you're going to travel by your vehicle, or maybe on foot, right? So this is the path. They will try to cross this path, right? And based on the tracks that are left by these animals, especially large size animals, because they have to be heavy so that they can make tracks, you can identify the species, and you can also understand the abundance of the species in that particular jungle. So you need to understand the tracks, you need to be able to identify the footprints, and based on that you can also understand the age of the animal, you can also understand the population of the animal, and you can also understand how much time it is spending in that particular area. So this becomes observational foot track survey. So this is the track, these are the footprints left by these particular animals. Now we'll move to so this was all direct way. Okay, so here you are directly looking at the biodiversity. Now as we move forward, this becomes the indirect way of understanding biodiversity. So you go out into the forest and you look for pellets, okay, and you look for the scat, and by looking at the scat you can understand the activity, you can understand if the animal is suffering from some disease, you can understand if it is in heat, you can understand what it is eating, you can understand if it is carrying some pathogen, you can even understand the sex. Individually you can identify that particular individual, right? So by looking at the excreta left by animals, right, indirectly you can get the diversity, richness, you can get the abundance of these particular animals. Okay, now next is next method is audio monitoring, right? So if you must have learned that frogs and birds, they sing or they make sounds, right? So what we do is we have some instruments, right, and these instruments we place them into the field, we place them in areas where we know we can find frogs, right, especially near water bodies, ponds, and we leave it on for some particular number of days or during the breeding season, say during the breeding season, and we record the calls that are made by these organisms. So when we record the call, we go back when we finish off with the work, we take this instrument, we take the chip out of it, we put it into the computer, and we try to analyze the different kinds of sounds produced by the different species. So you already have a standardized sound database with you. You compare the sounds created in that particular area by these animals, and what you do is you try to compare what animals are present in your area just by recording your calls. Now, if you go back to the direct observational ways, here you have to come every time to check what kind of animal was present in this particular quadrant, or you have to walk from year to year, and every day you every time you have to come, every time you have to go through this rough terrain, you have to stand here for long periods of time, record, do the observation, record the calls. Here you have to map the entire territory. Here you have to very intensively search for nests, right? Go into areas where you cannot actually reach very easily. But here it becomes very, very easy. Just once you have to put this instrument and it can record the calls, say for 15 days or for even 30 days. You just go back the next time, take out the instrument, bring it to the lab, and you can just sit in the lab and get the number of species that you can find in that particular area. Okay, next thing is called as camera traps, right? So these are very extensively used by the forest officials to catch hold of predatory large mammals, okay, or even large herbivores which occur in dense forest. Okay, so every time you cannot get into the forest, every time you cannot go and disturb the habitat, so what you can do is you can just put this camera into the forest, which has got a infrared sensor. So if an animal passes nearby and if it comes within the range of this sensor, because of the body heat, right, this sensor can see the animals that are moving, and once it detects the animal, this digital camera will click the photograph and store it into it for say 1 month or even 2 months. Okay, so you can come back after 1 month. It can even record videos for short period of time. It can even record images and videos during the night time. That's why we have a flash which the animals cannot see, and it can help you to record even the rarest events which occur in nature, which normally you will not be able to see if you go into the forest. And as I told you, you can keep it for weeks or you can even keep it for a month, and it will continuously keep on taking images of the animals that cross path within the range of this infrared sensor. Okay, right. So now things have become so advanced that these camera can give information to satellites, and these AI enabled cameras are sending out real time alerts to the forest officials. So if there's a tiger roaming around and it comes near a particular village, the forest official will get warning that this tiger has entered into a particular territory, and they may shoo it away back into the forest, or they may warn the villagers that this tiger is sure to come into your area. Okay, and even as I the aim of this particular topic is biodiversity assessment. This has been extensively used. The camera trap survey has been extensively used to monitor the tiger numbers in India, and this has entered into the Guinness Book of Records as well. Okay, but sometimes it has got a negative side to it. Just a few days back, on 29th November, there was a news where these camera traps were used to peep at women, and this was a study that was done by the Cambridge University, and this has become a very controversial issue. So the thing is, uh, technology can help us to ease our burden of understanding diversity and abundance, but it can also lead to such kind of problems. Okay, now moving forward, now we move to the second kind of category. The first category was observational method, right? Observational sampling method, right? The next category is going to be where we actually capture the animal to look at the diversity. Okay, so we can't go and catch each and every organism. So what we do is we construct a pit, or we construct a trap. We fill it with some chemical, or we fill it with some pesticide, and we leave it. We protect it with some canopy so that rainwater doesn't enter inside, right? We cover it with some leaves. We put leaves inside. The unaware insect or animal will just walk and fall into it, and because of the preservative will get preserved into this and will not rot, right? So if you don't have a preservative, the animal may rot. What we do, we use a preservative inside, and anything that is trapped inside we collect, go to the laboratory, and we try to identify it under whatever means, microscope, or by using our methodologies that we have. This can be used for insects, this can be used for reptiles, amphibians, and even for small mammals. Okay, so very commonly used method called as the pitfall method of capturing animals to understand the diversity and also the abundance in a given area, right? The thing is, you just can't put one trap, you have to put many traps so that you can get an idea of what organisms are present in that given area. Now if you're looking at small mammals, you can also go for something called as snap traps, okay, or simply we call them as rat traps. They will catch hold of the animal, and you can study them, and you can again leave them back into the nature. Okay, right. Then we have something called as funnel traps, which we use for flying insects. Okay, so if you go here, this was mostly for insects that are in the soil, which crawl on the ground. So if you have insects which are flying, you can attract them using some methodology, say light or some chemical or some hormone, and they will get trapped into this particular trap. So they are funnel shaped. That's why they're called as funnel trap. So you gather them here, and after some say some 15 days or 10 days, you can just take